Okay, thank you very much for introduction. And thank you very much, Andrew and other organizers for giving me a chance to present this talk at such a large audience. I didn't expect that large, but I would like to report one of our recent studies of hue selectivity of brain activity using one of the EEG technique called SSVEP. And this is a collaboration with authors listed here. And let me start with the first slide. This is a overview of the color representation. As you know, that the color representation starts with three types of cone photoreceptors, and it is converted to the cone opponent responses in retinal ganglion cell, and it will be transferred to some kind of signal, and then finally it reaches to the categorical kind of signal in the brain for the communication or memory purpose. So have been interested in how the color signal is transformed during this post-opponent system in the brain. And I would like to introduce some of our recent struggles about this phenomenon, issue. And one of our studies we have conducted with fMRI was to identify the number of voxels which is selective to each individual hue. So here is a result of a study published in about 5 years ago, which is identify the hue selectivity of voxels, each voxels by using the bold fMRI technique. And three figures on the top, three panels on the top is showing the result from three different participants in primary visual cortex. And in the radio plot, each of which is showing the population of hue selective voxels in by the distance from the center of this chart. And horizontal and vertical axis is showing the direction of L versus M opponent selective direction, and the vertical axis is showing the S cone selective direction. So what you can find with this diagram is very strong individual differences, but another characteristic is that it shows a very strong anisotropy. It has abundant voxels preferring to blue and yellowish directions in each subject, but you don't find much voxels which is selective to the red-green direction, particularly along the L and M cone opponent axis. So, but it is known that there are a lot of color-selected neurons in the electrophysiological studies in animals. So this could be just a part of the tip of the iceberg, and we are missing some submerged part of the brain activity responding to other kind of cells, which should be more uniformly distributed in the visual cortex. And one of the probably reason is because the size of the voxels we used for the recording was quite huge, 2 millimeter, 2 millimeter by 3 millimeter, compared with neuron. This is very large. So probably only overpopulated neurons within contained one voxel could have emerged. We could just measure that kind of response as a hue selectivity of the voxel. So another, oh, sorry. I don't know why it is not moving. Okay, so in order to overcome this issue, we introduced our EEG recording technique called SSVEP, which is a steady state visual evoked potential, to see whether we can record a much more broader distribution of the hue selectivity in the cone opponent space. And these are the collaborators, Sae and also Soren Andersen from UK. And Sae also mostly conducted this experiment in this study. So the stimulus was like this. This is almost the same stimulus setup used in our fMRI study, which was a checkerboard pattern alternating its color between color and gray in at 5 Hertz in time, so that evoked potential will take a 5 Hertz component corresponding to the checkerboard flickering alternation of the checkerboard. And the color was changing. Meanwhile, along the hue circle defined which is defined in the LM and S-con opponent space. I will introduce this later more in detail, but the hue was changing gradually by taking 24 cycles per cycle. And the subject was fixating at the center of the screen provided by the fixation cross. And meanwhile, subject was conducting a go, no go task. Either circle or cross symbol was presented randomly on the screen in the area of visual stimulus, and subject was asked to pay attention uniformly across this stimulus area, and they were asked to press a button when they found a circle symbol, but not press a button when they find a cross symbol. And in order to present this visual stimulus, we use the Visage system, and the brain activity was recorded by brain amp system, EEG system. And the color stimulus was like this. It was defined on the cone opponent space and under Isochromal defined for each subject by flicker photometry. And we used three different contrast levels of the hue circle. One is a full contrast condition, which is the maximum extent we can present on our display. And the second one is the half of it in terms of the cone contrast with respect to the cone activity for the equal energy white, which is the origin of this space. And the quarter is just a quarter of it. And the scale of the LM and S cone axis were roughly equated by the multiples of threshold. And the recording for each contrast was repeated 24 times by changing the starting point randomly across this circle so that any kind of artifact by the modulation within the cycle will affect the result. And also we tried clockwise and counterclockwise direction color changes to confirm that there is no hysteresis effect in the result. And here is the spectrum of SSVEP. You can find a 5 Hz component, which is strongly responding to the color stimulus. And you can find different colors, red, green, and blue here, which is corresponding to the full contrast, half contrast, and quarter contrast. And if you average the amplitude of this component from 16 subjects, I think, you find that the amplitude was changing in proportion to the chromatic contrast of the color stimulus. And now I will show you the hue selectivity profile of the EEG response. And this is a diagram. Again, horizontal axis is showing the L versus M cone opponent axis, and vertical axis is showing the S cone selective response direction. And the three lines here, contours here, are showing the difference between full, half, and quarter contrast conditions, showing that the EEG responses were nicely changing in proportion to the stimulus strength under Isochromal. So this could be something surely responding to the color stimulation. And now you can find that the trajectory here is, this is a result of the average across 16 participants, and the shaded area is the standard errors of mean, but you can find that the elongation is strongly found in this magenta green direction, which is not a simple line symmetric shape that could be predicted by the cone opponent based response. So in order to confirm which kind of underlying mechanism could evoke this kind of hue selective profile, we checked, tested the model fitting by assuming 3 types of models, different models, and its combination. And I would like to explain detail one by one, but one is a cardinal model. This is the simplest one, which is assuming that the response to the particular hue is the vector summation of the LM component and S-axis component in as a vector sum. And another one is a scalar sum pattern, which is assuming that the response to the one particular hue will be the sum of the LM component and S-cone axis component. And here's a definition. So it's just simple. By why we assuming this is because EEG recording, EEG technique is recording the electric potential at the scalp and in additive manner. So if different kind of electric source is activated at the same time, the potential that emerges at the scalp will be just a simple summation of these electric potentials. So we can assume that the amplitude will be the sum of M component and S component. So the expected trajectory would be like this. And another model we introduced was a Mansell isochromal locus, which is based on this Mansell isochroma contour calculated from spectral data of Mansell color chips provided by University of East Finland, and which shows a nicely fit by a symmetric shape. It's actually a ellipse centered at 0, 0 here. And our stimulus profile was, like this. So the full contrast condition, the extent of stimulation was like this, and half contrast was like this. Since this locus is showing the isochroma contour, it means that a strength stimulation in color contrast in this direction, magenta green direction, was too strong compared with this direction, cyan orange direction. So you'll be able to predict that a response based on this isochroma mechanism would be elongated in this magenta green direction like this. And we checked which model can best fit our data by isochroma control model or cardinal model with vector sum and scalar sum model and its combination with isochroma contour. And it turned out that the isochroma contour model with cardinal scalar sum model best explained our data. And if you are interested in this result, you can find a detailed description in our paper, which was published in cerebral cortex communications, which is online issue of the cerebral cortex, and it's a open access journal, so you can find it if you like. Please check the paper if you like. So what kind of mechanism could evoke this kind of isochroma response? So one of the possible candidate is a multiple channel model, which has been proposed and studied in psychophysics for many years. And here is the plot, which was taken from Emery's study in 2017, which was derived by factor analysis of elementary color scaling. And it shows a nice narrow-tuned hue selective mechanism. Actually, it's 7, I think, which is covering entire hue circle. And whether if the Mansell isochroma-like response was evoked by this kind of mechanism, we might be able to achieve this kind of individual hue selective mechanism by SSVEP data. So the next part, I would like to introduce our study or struggle to find that kind of underlying mechanism. The collaborators, again, Sai did main work in this project here, and David Petozzo is helping us in analyzing the data. So the individual differences in SSVEP was like this. I just presented grand mean data in the previous slide, but the individual data was very, very different in its shape of the hue selectivity profile. So if the common mechanism is underlying this differences, just a gain or a population is different between individual, you might be able to derive that kind of underlying mechanism by analyzing these individual differences. That is the basic idea of our attempt. And introducing factor analysis is one idea, but we began the analysis with a much more basic analysis in this project. So this is a plot showing the 0-degree stimulus, which is a plus L direction stimulus, and its amplitude from 16 participants as a horizontal axis, and taking the same 0-degree amplitude at the vertical axis, you will get this kind of line-shaped distribution, which has a correlation coefficient of 1.0. And by holding this horizontal axis component and changing the vertical axis component by 15-degree step, by the way, we average the result of the amplitude by 15-degree beam, and we use this that number as a vertical axis for plotting in this small scatter diagram. And if you change the vertical axis gradually by 15 degree, 15, 30, 45, and 60 degree, you find that the correlation becomes almost zero. It has no correlation between 0-degree amplitude and 60-degree amplitude. And if you progress more and more, and around 1 or 5, you will find a negative correlation, which is significantly negative, when you take the 0-degree amplitude in the horizontal axis and 105-degree as a vertical axis. And it goes back to the zero correlation again, and you again find a strong negative correlation in 240 degree when the horizontal axis was 0-degree amplitude. And we checked the individual differences in this way. And if you vary the horizontal axis component in the previous slide gradually by 15 degree, you can finally obtain this correlation coefficient matrix, which subtends 24 by 24 elements. And the top row is actually the same as the plot I have shown in the previous slide. And in the first glance, you can notice that there is a knot-like structure, blob-like component along the diagonal line, which is meaning that some of the hues were captured by the same mechanism. I mean, as you change the hue, the pattern of activation is not changing. That is what this diagram is meaning. So there is some limited number of structures seems to exist in these responses. And also you can find the negative and positive correlations, which is shown by the brightness of this color code here. Dark one is corresponding to minus 1.0 and bright one is corresponding to 1.0. So for example, you can find a dark, bright, dark, bright, dark, bright, this kind of periodical change of the correlation changes can be found at around 120 and 240 degree. Maybe it is not so obvious to find the vertical alignment of this component, but if you align this diagonal component in vertical like this way, you can now find the streak which is in oblique around 45 degrees. So again, the vertical axis, the off-axis component, which has a periodical changes of correlation, seems to be aligned in the vertical line like this. And here is showing the correlation coefficient matrix with these 4 dotted lines are showing the direction or the location of the cone opponent axis, which is already known mechanism as a retinal ganglion cell system. And apparently the bright dark spot repetition is of this cardinal axis mechanism, which is seen to be something else of the cone opponent mechanism. And another clear bright dark repetition can be found around 0 degree or 180 degree, and probably these two are corresponding to the L minus M or L versus M system. And let us take a look at this 120 and 240 system more in detail. This is a diagram is showing the same result in the just a hue circle chart. And when the 0 degree component shows a 1.0 correlation, 120 and 240 degree system, I don't know what they are, but it's showing the strong negative correlation. And when you switch the hue of attention to 120 degree, for example, and it shows a strong 1.0 correlation, and 0 system and 240 system shows a 0 degree system shows a strong negative correlation, and 240 degree shows a mild positive correlation, and 180 degree seems to show a weak negative correlation. And 240 degree again shows strong when the 240 degree system shows a positive correlation. 0 degree shows negative, and 120 shows a weak positive correlation. That is what we have found so far, and it seems to be different from the 4 cardinal mechanisms. And these 2 mechanisms are probably corresponding to LM, but we don't find something that kind of repetitive changes in this area, 60 degree and around 300 degree. But when we get back to the finding that the not-like component is found on this diagonal line, this 360 degree system seems to exist, but seems to be behaving in a different way as 120, 240 degree or 0 or 180 degree system. And this is just a result from full chromatic contrast experiment, but you can do this kind of analysis for half and quarter contrast result. And this kind of further analysis is just now underway, and we would like to present the details in future. We have submitted abstract to VSS this year, so we hope we can report much more detailed and further result at this in the future meeting by this author. And now another our interest is that what are these not like system? I mean, if these are some hue selective, narrowly tuned hue selective mechanisms, can we measure this by masking or some kind of other technique? So the last part of my talk is about a hue selective masking experiment, which is conducted by Anya again. She's supervising our undergraduate student, Saki. And they were doing the recording by using SSVEP masking technique. Now we present the mask color in addition to the test hue, which is changing along the hue circle by taking 24 seconds, like the first experiment. The test stimulus was flickered at 5 Hertz, and the mask stimulus was staying at one of these 4 hue during one trial, but it was flickering at 8 Hertz. So if you monitor 5 Hertz and 8 Hertz, you can identify which part, which component is corresponding, which component of EEG is corresponding to the response to the test and the mask stimulation. And this style is inspired by the Peltazon-Nocia study in 1997 on special frequency tuning experiment. And here is the EEG spectrum from one particular subject. Different panels show the different mask conditions: 45 degree magenta, 135 cyan, 225 green, and 315 orange mask conditions. And all the mask conditions shows nice 5 Hertz component corresponding to the test stimulation, test color stimulation, and 8 Hertz is corresponding to the mask color stimulation. And by measuring the no mask condition, the test only response without presenting any kind of mask color, we use this as a standard amplitude to normalize these results to obtain the masking effect. And here is a masking effect shown as a function of the angle of test hue relative to the mask in the horizontal axis. And the vertical axis is a log normalized amplitude of the test response of the test stimulus. It shows a bottom or dip at around the 0 degree around the mask hue, and it seems to show some peak offset from the 0 degree by some in sideway, depends on the hue of the masking stimulus. And here is the masking effect profiles from 19 subject, average of the 19 subject, and different panels show the result from different masking conditions: 45 degrees magenta, 135 cyan, 225 green, and 315 degree orange masking condition. And it seems like the bandwidth of the masking effect seems much more broader than we were expected at the beginning. And if we fitted this curve by a circular Gaussian function, the half width, half maximum was about 60 degree for 45-degree condition. And 135-degree condition shows slightly narrower bandwidths, but it showed very large bandwidths than we expected. And again, we tested whether this this result can be modeled by the cardinal model prediction. Again, this is a vector sum model, which is assuming that the response to a particular hue is a vector sum of the horizontal and vertical component. And the black line here is showing the prediction in ideal condition and no mask without presenting the masks. And when the mask was presenting, for example, the 45-degree, two systems corresponding to the positive direction of L and positive direction of S is response is reduced, and the predicted response would be like this line shown in the red line here. And the scalar sum model can also predict a similar kind of profiles. This dotted line, black line here, is showing the condition without mask, and the red line is showing the condition with a mask at 45-degree in this case. And by taking the logarithm of the each response and took the difference, you can obtain the predicted function shape of the masking function. And very significant characteristic of this predicted curve is it has a very wide flat bottom, which ranges at the entire quadrant spanning -45-degree to 45-degree. And it doesn't seem to fit our result. I mean, they don't fully account for masking effect profile. So some cardinal mechanism and additional hue selective model, again, may be needed to fully account for our prediction. Sorry, I'm running out of the time, so I'll just skip this, just go quickly, but again, it doesn't show a flat bottom in our result. And in summary, we recorded the SSVEP response to isoluminant hue to find that elongated profile, which was nicely fit by the Mansell isochromal contour in combination with the cardinal response, which is shown in the details are shown in this paper. And individual differences are analyzed and finding some new system probably, and the masking experiment is underway. And I would like to note that this second part will be reported at the VSS, hopefully, if accepted. And finally, I would like to send my acknowledgement to my collaborators and funding support. And thank you very much for your attention. That's it.